The adult spinal cord measures about 18 inches in length or 45 centimeters in length and has a maximum width of about a half inch or 14 millimeters. The cord itself is not as long as the entire spine, and the cord ends at about the levels of L1 and L2 vertebrae. The posterior or dorsal surface of the spinal cord has a shallow groove called the posterior median sulcus that we can see here on the cross section. The anterior or ventral surface of the spinal cord has a wider groove called the anterior median fissure. In the cross section, we can also see the distribution of the gray matter versus the distribution of the white matter. The gray matter of the spinal cord is enlarged in areas that are dedicated to the control of the limbs. These enlargements are termed the cervical enlargement that supplies nerves to the shoulder and upper limbs, and the lumbar enlargement that provides innervation to the pelvis and lower limbs. The spinal cord starts to taper and becomes conical just below the lumbar enlargement. This region is termed the conus medullaris. A slender strand of fibrous tissue called the filum terminale extends from the inferior tip of the conus medullaris as far as the second sacral vertebrae, where it provides support to the spinal cord and is a component of the coccygeal ligament. There are some variations in the proportions of gray matter versus white matter in the cervical, thoracic, lumbar, and sacral regions of the spinal cord. Here we can see that the lumbar region has the most amount of gray matter, and the thoracic region has the least amount of gray matter versus the white matter. The entire spinal cord can be divided into 31 segments on the basis of the origins of the spinal nerves. A letter and number will designate the spinal nerve beginning at spinal nerve C1 that exits the intervertebral foramen between the occiput and C1 and ending at the spinal nerve S5 that exits the fifth sacral foramen. Every spinal segment is associated with a pair of dorsal nerve roots and ventral nerve roots. The dorsal root and the ventral root will merge to form the spinal nerve just lateral to the intervertebral foramen. The dorsal nerve carries sensory nerve fibers and the ventral nerve root carries motor nerve fibers. The cell bodies of the dorsal nerve root are clustered in the dorsal root ganglion. The dorsal root ganglion is located between the pedicles of the adjacent vertebrae. Distal to each dorsal root ganglion, the sensory or dorsal and the motor or ventral roots are bound together into a single spinal nerve. Spinal nerves are classified as mixed nerves because they contain both sensory, or afferent, and motor, or efferent fibers. There are 31 pairs of spinal nerves, each identified by its association with adjacent vertebrae. Cervical spinal nerves are associated with spinal nerves that exit the cervical spine, and lumbar spinal nerves are associated with spinal nerves that exit the lumbar spine. Each spinal nerve inferior to the first thoracic vertebrae takes its name from the vertebrae immediately superior to it. Thus, T1 emerges immediately inferior to the T1 vertebra. T2 emerges immediately inferior to the T2 vertebra. This arrangement continues all the way down through the lumbars and into the sacrum. The L4 spinal nerve exits just below the L4 vertebrae. This arrangement is different in the cervicals. The first cervical spinal nerve, which is C1, passes between the occiput and the first cervical vertebrae. Thus, the C1 spinal nerve exits just superior to the C1 vertebra. C2 exits above C2. A spinal nerve designated as C8 will exit just below the C7 vertebrae. Therefore, although there are only 7 cervical vertebrae, there are 8 cervical nerves. This segment is on the spinal meninges. The spinal cord and its associated tissues are protected from damaging contacts with the surrounding bony walls of the vertebral column with a specialized layered membrane called the spinal meninges. The meninges provide physical stability and shock absorption. The spinal meninges consist of 3 layers: the dura mater, the arachnoid mater, and the pia mater. At the foramen magnum of the spine, the spinal meninges are continuous with the cranial meninges which surround the brain. A bacterial, viral, or fungal infection can cause meningitis or inflammation of the membranes of the meninges. The dura mater is a tough fibrous layer that forms the outermost covering of the spinal cord. Between the dura mater and the walls of the vertebral canal is the epidural space, a region that contains areolar tissue, blood vessels, and adipose tissue. This is the site for epidural anesthetic injections. The dura mater will taper to a dense cord at the end of the spinal cord and will blend with the components of the filum terminale. The middle meningeal layer is called the arachnoid mater. The space between the dura mater and the arachnoid mater is termed subdural space. The space between the arachnoid mater and the pia mater is termed the subarachnoid space. The subarachnoid space is filled with cerebrospinal fluid, which acts as a shock absorber and a medium to distribute dissolved gases like oxygen, as well as nutrients, waste products, and chemical messengers. A spinal tap is a procedure where a needle is inserted into the subarachnoid space and fluid is withdrawn to be studied for infection and other disorders. The pia mater is the innermost layer that is closely adhered to the spinal cord. The pia mater consists of a meshwork of elastic and collagen fibers that are firmly bound to the underlying neural tissue of the spinal cord. The spinal meninges also cover the dorsal and ventral roots as they pass through the intervertebral foramen. The meningeal membranes are continuous with the connective tissues that surround the spinal nerves and their peripheral branches. This segment is on the organization of white matter and gray matter in the spinal cord. The anterior median fissure and the posterior median sulcus divide the spinal cord into left and right sides. The white matter is more superficial and contains large numbers of both myelinated and unmyelinated axons. The gray matter is dominated by the cell bodies of the neurons, neuroglia, and unmyelinated axons. The gray matter surrounds the narrow central canal and forms a butterfly or H shape. The gray matter on each side of the spinal cord can be divided into three regions called horns. There are the posterior gray horns, anterior gray horns, and lateral gray horns. Each of these horns contain different nuclei. Nuclei are masses of gray matter within the central nervous system. There are sensory nuclei that receive and relay sensory information, and motor nuclei that issue motor commands to the peripheral effectors. The white matter on each side of the spinal cord can be divided into three regions called columns. There are the posterior white columns, the anterior white columns, and the lateral white columns. The anterior white columns are connected by the anterior white commissure. Each column contains tracts whose axons share functional and structural characteristics. Ascending tracts carry sensory information toward the brain, and descending tracts carry motor commands to the spinal cord.